I've got a note saying my speaker is not working properly. Can you guys hear me?
We can hear you.
Great. Before we begin, are there any questions? All right, if there's no questions, today is August 22nd, and we are supposed to be working on chapter 13. We get there? Sharing my screen now. It's not giving me the option to move where we last were. All right, I have that we talked last time about latent and persistent viral infections. Can somebody confirm that?
Yes, that's correct.
Thank you. All right. Are there any questions about viruses at all? Because that's actually the last slide discussing viruses. All right. If not, let's move on to prions. Prions are proteinaceous infectious particles. When I first started talking about them, I was uncomfortable discussing prions because they're so different. They're a protein that causes a disease. And it's very unusual to have a protein cause a disease. But they are infectious proteins. They can be inherited. They are transmissible by ingestion, by transplant, and surgical instruments. By transplant, that's usually from a organ transplant. Prions cause spongiform encephalopathy, which means vacuoles in the brains. This happens in humans and animals. Let me show you vacuoles in the brains. There we go. There's a normal brain, and all of these are prion-affected brains. This one by Kuru, this one by classic CJD, what is that called? Creutzfeldt-Jakob disease, and this one by Scrapes. It's spongiform because it makes the brain look like a sponge. Any question about any of that? All right, in animals, the most well-known and probably the most common spongiform encephalopathy is bovine spongiform encephalopathy, more commonly called mad cow disease. But prions also cause sheep scrapies, cervid chronic wasting disease, and a disease in elk in like southwestern United States too. In humans, we don't call it this or even spongiform encephalopathy. They're called Creutzfeldt-Jakob disease after two doctors who discovered it, running in two families, or Gerstmann, Strassler, Schneckner syndrome, and I could be really butchering that name because I'm not German. And they found it in other families. And initially they thought it was a different disease, but it's actually a similar disease. And then fatal familial insomnia, and then Kuru. So all of these are human spongiform encephalopathies. Any question about any of them? All right, let's talk about Kuru. This disease was seen in the Fore tribe in Papua New Guinea. There's Papua New Guinea, and the red is where the Fore tribe is. When the patient became sick, they became unable to walk. There was the loss of the ability to swallow or chew, drastic weight loss, and it led to death. How was it transmitted? In the Fore tribe, they had an unusual funeral ritual where people, when they died, were cooked, and then their body was eaten by their relatives. So the dead were actually eaten by the relatives. Now, fortunately, the mothers in the Fore tribe noticed that their children, as well as they, were getting sick after eating their dead, and so they changed their behavior and refused to have their children eat their dead relatives, and they stopped eating their dead relatives as well. So when the people, the white people, Europeans, came to Papua New Guinea, this disease was only seen among the men in the Fore tribe, and the men still ate their dead relatives. And then the missionaries came in, and they were totally, oh, what's the word I'm looking for? Totally upset about this practice, and they put it into it, saying this is what's the word I'm looking for? Just totally unacceptable. And I don't know when this was, in maybe the 1920s to the 1940s, and the missionaries passed laws, not really among the Fore tribe, but among the nation of Papua New Guinea, that you could no longer eat your dead relatives. Okay. And anyone who did, of course, would be, imprisoned or put in jail or something like that. And so there were penalties. And take it as you wish, the missionaries are often stated as being non accepting of different cultures and their customs. But in my opinion, this is only my opinion, they did a good thing for the Fore tribe because the missionaries got rid of Kuru in the Fore tribe. And so they actually did something good in their intolerance of the rituals of the Fore tribe. Any question about any of that? All right. Prions are a proteinaceous infectious particle. How the disease comes about, meaning the mechanism of the disease, is there's a normal protein called PRP superscript C, which is a normal cell surface protein found on nerve cells. And this protein needs to be there for the nerve cell to function. And when the normal cellular protein, PRP superscript C, comes into contact with the prion form of the protein, PRP superscript SC, SC standing for scrapies. The scrapies protein can transform the shape of the PRP superscript C protein into the shape of the PRP superscript SC protein, or the scrapies protein. And the only difference between the PRP superscript C and the PRP superscript SC, meaning the prion protein, is the folding of the protein. They have the same amino acid sequence. Any question about any of that? And how the prion protein replicates is it simply comes up to the normal protein, causing it to change its folding pattern into the prion protein. The cell then says, oops, I don't have enough PRP superscript C protein, the normal protein, so it makes more. The trouble is there's now two Scrapes proteins in the cell, and if one of them comes up to the PRP superscript C protein, it folds it into the prion protein, or the Scrapes protein. And then the cell makes more of the normal protein. The Scrapes protein just continues to accumulate in the nerve cells, especially in the brain cells, forming plaques and eventually leading to the nerve cell death. Any question about any of that? Well, there is one question you should ask, and that is, how did the PRP superscript SC protein first come about in the nerve cell? Well, it's thought that every once in a while, by mistake probably, the PRP superscript C protein can convert to the prion protein. Very rarely does this happen because prion disease is not usually found in either animals or in people. But every once in a while it does. And then when you get one prion protein in the nerve cell, it can come up to more of the normal protein and convert it into the prion protein. In the families that have prion disease, and that's largely the Creutzfeldt-Jakob disease, but it could be the other disease as well. The family has a form of the normal protein, which is more likely to convert to the prion protein. And this conversion doesn't happen until the patient is usually in middle age, meaning when they're young, they have the normal protein and then they continue reproducing, and then they're young, get born with this, we should say, variant of the normal protein, which is more likely to form the prion protein. But that variant only converts when the individual becomes at least middle-aged or elderly. And that's how the disease runs in the families. The people get prion, but they only get prion after they've reproduced. Any question about any of that? All right, let me back up just a bit and say the most well-known case of a prion disease was when in England they had a epidemic of mad cow disease. And what happened was one of the cows got prion disease, and the British farmers, or the ranchers, I guess you could call them, wanted to sell the cow brains, but nobody wanted to buy them. So the British had the habit of taking cow brains, grinding them up, and then adding it to grain, and then feeding the grain to cows. And one cow got prion disease, and the brain was ground up, fed or put in the grain, fed to cows, and then many cows got prion disease, and then this just ballooned as more of the cows brains were slaughtered, the cows were slaughtered, and the brains were ground up, added to the grain. And then there's a whole epidemic of mad cow disease in Britain. The British finally got control of the situation by incinerating, meaning killing and incinerating, all of their cattle, and then importing cattle from other countries to resupply their depleted herds. And that's the only way they could finally get control of the prion disease. Before they grabbed control of the prion disease, I do need to mention that about 150 people came down with prion disease, most likely from eating prion-tainted beef. and then about 150 British cats, both domestic kitty cats and zoological cats like lions and tigers, came down with prion disease. Surprisingly, dogs did not, even though they were eating about the same food as the British kitty cats. So the dogs must have been more resistant to getting prion disease when they consumed it. The cats were not. Any question about any of that? All right. Let me state that scabies disease is a disease that's been affecting sheep in Europe for centuries. And sheep farmers, especially in Northern and Eastern Europe, have been dealing with scabies disease for a long time. Any question about scabies or prions, I guess? All right. If not, let's move on to talking about plant viruses and viroids. Plant viruses resemble animal viruses in many ways, and they cause diseases of economically important crops, such as beans, corn, sugarcane, and potatoes. The only plant disease I know is tobacco mosaic disease, which causes disease, obviously, in tobacco plants and then related plants like tomatoes. Actually, there is a, no, that's not a plant disease. Sorry, that's a plant virus. That's a um, fungus-like protus that I was thinking of. And then there's also the viroid disease. Those are the only other plant diseases I know. So that's the only plant virus I know. Because of the tough cell wall of plants, the virus tends to enter through wounds in the plant via a parasite, such as a nematode, an insect, or a fungus that damages the plant cell. And then the virus can enter. Infected plants can then spread the virus in either their pollen or their seeds. With tobacco mosaic virus, the virus can get on somebody's hands, like if they're a smoker, and then the person touches the tomato plant, the virus can move from their hands to the tomato plant. I just happen to know about that one. All right, I'm not going to talk about plant viruses any further because these are viruses that do not infect animal cells and they do not cause human disease. And the focus of this class is discussing microbes that cause human disease. Any question about any of that? All right, let's move on. Let's talk about viroids. Viroids are a naked RNA molecule which is infectious. You can think of it as a naked RNA virus lacking the protein coat, meaning it's only a molecule of RNA. Viroids are plant pathogens. And here you can actually see a viroid causing disease in plants. And one disease is the potato spindle tuber viroid. Let me see if I can get that. Where the potatoes are truncated. I should have a picture of that, but for some reason I don't. Oop. There we go. The potatoes on the right are potatoes affected by the potato spindle tuber viroid. The potato's on the right. The potato on the left is a normal potato. Now, this is a better example. Much normal, big potato, not affected. And these are potatoes affected by the potato spindle tuber viroid. Any question about any of that? All right, there are no known animal viroids. There are only plant viroids. Any question about anything? If not, that's it for chapter 13. Let's move on to chapter 14. Oops, wrong one. All right, chapter 14, Principles of Disease and Epidemiology. This slide is showing you the major goals and a rough outline. Know the terms, know pathology, etiology, microbial antagonism. And in the terms in this chapter, if all you do is learn the terms, you will learn 50% of the material in this chapter. So terms are extremely important in this chapter. Any question about any of that? All right, 2, understand the relationship of the normal microbiota and its host. 3, understand the stages of disease development. And 4, understand nosocomial infections and how to control them. Any questions about what we're going to cover? All right. Some terms you should know. Pathology is the study of disease. Etiology, the study of the cause of a disease. For example, the etiological agent of the disease anthrax is Bacillus anthracis, the microbe Bacillus anthracis. Pathogenesis is the development of a disease. Further in pathology, infection, and disease, infection is colonization of the body by a pathogen, or it could be the presence of a microbe where the microbes are not normally found. For example, E. coli is normally found in your gut, but if it is found growing elsewhere, like in your kidneys or in your lungs or in your ear, it would cause an infection. It is not supposed to be growing in any of these places. Disease is an abnormal state in which the body is not functioning normally. When we're talking about disease, you should come to realize that only a few microorganisms can cause disease. Only a few are pathogenic. Some microbes are, in fact, beneficial to the host, where they give the host some benefit. These are largely, but it may not be entirely, the microbes of the normal microbial system, the normal microbial ecology living with and in on us. Any question about the normal microbiome? Not all the normal microbiome benefit us, only some of them. But all animals and plants have large communities of microbes found in association with them. And this we call the normal microbiota. And then the individual or plant boosting the normal microbiota is the host. You can have a human normal microbiota and then something like a kitty cat normal microbiota. And they will differ, at least on some organisms. Of course, cats are maybe not the best example because they have a higher body temperature than humans. But one normal microbiota of cats is Bartonella henselae, which causes cat scratch disease in people, meaning it actually is not part of our normal microbiota. And if humans get it, it will cause an infection that we call cat scratch fever. Any question about any of that? So there is a human normal microbiota and then different normal microbiota for each different species. The normal microbiota are microbes that essentially permanently colonize their host, meaning shortly after I was born, and this is true for every human, I got Staphylococcus epidermidis on my skin. In fact, this one I got probably during birth, picking it up from my mother. And that's probably true of everybody. This is a part of human normal microbiota, Staphylococcus epidermidis. And once I got it, it will permanently colonize my skin for as long as I'm alive. The normal microbiota does not have to be permanent, but essentially permanent, meaning for a very, very, very long time, for most of the individual's lifespan. The normal microbiota is only found in certain regions of the body. And then disruption of the normal microbiota and the host balance with the normal microbiota appears to be associated with disease and impaired immune function. For example, if you were to take antibiotics, that would disrupt the normal microbiota host balance, killing off large numbers of the normal microbiota, which frequently gives the patient either diarrhea or constipation. Fortunately, the normal microbiota host balance reestablishes, and then the diarrhea or the constipation goes away. Any question about any of that? All right, this picture is showing you the locations of the normal microbiota. Found on the skin, obviously. Also the openings in the face, such as the mouth, the eyes, the nose, the throat. The intestines, especially the large intestine, but all the intestines, as well as the stomach, have a normal microbiota. So down the gastrointestinal tract, we have normal microbiota from the very beginning all the way through the tract to the very end. With the nose and the throat, only the upper respiratory system has a normal microbiota. Lower in the respiratory system, especially the lungs, does not normally have normal microbiota. And if ever we have microbes growing in the lung, it would cause pneumonia. And then the lower urinary and reproductive system, such as the urethra in both sexes, the vagina in females, and it's not stated here, but also the bladder in females, have a normal microbiota. And as I stated, I don't know if males have a normal microbiota in their bladder or not, because nobody's ever done research, the last I checked, which admittedly has been a year ago, to see if males have a normal microbiota in their bladder, but females do. So that's where the normal microbiota is normally found. It is not normally found in the blood, cerebrospinal fluid, the muscle, the nervous tissue, the bone, anything internal to the body and not open to the outside. Microbes are not found there. Any question about any of that? That is the normal microbiota. There can also be a transient microbiota, where a microbe can be present for days, weeks, or months, but unlike the normal microbiota, it does not permanently or essentially permanently colonize the host. I once had a transient microbiota, and that is I went and visited my aunt, and she had a Staphylococcus aureus skin infection. And of course, she insisted on hugging me and my mother who were coming to visit her. I don't know about my mother because I didn't test her. And then I routinely was testing my skin for Staphylococcus aureus about every 3 months at that point. And I never was positive for Staphylococcus aureus, except immediately after I hugged my aunt, where Staphylococcus aureus was growing and present on my skin. And I said, geez, must have got this from my aunt. And was going to watch it carefully. The next time I sampled my skin, and all future times after I sampled my skin, which was about 3 months later, I no longer had Staphylococcus aureus on my skin. So that was a transient microbiota, present for days, weeks, maybe even a couple of months, but not longer. Any question about any of that? And I was quite pleased that I was able to throw off Staphylococcus aureus. If you don't know, that is a human pathogen. And if you get skin boils and other things like that, you probably have Staphylococcus aureus growing on your skin, causing the boil. And only about 20% of Americans have Staphylococcus aureus on their skin, and they were much more likely to get boils and other skin lesions than the rest of us, 80%. Any question about the normal microbiota or transient microbiota? The normal microbiota is about 4 times 10 to the 13 bacterial cells on a person, on or in a person. The human has about 3 times 10 to the 13th body cells, meaning there are more normal microbiota of bacteria than there are human cells. And that begs the question, are we more microbe than we are human? And don't let that freak you out too much, because the answer is we do have more microbes than we do have human cells. Any question about any of that? Oh, this isn't quite right anymore. It's not 10 times anymore. They thought it was initially 10 times more, but actually it's only, what is that? About 1/4 more cells, maybe 1/3 more, something like that. Here's some picture of the normal microbiota, the cocci and the nasal epithelial, orange cocci. Here's bacteria lining the stomach, and these look like spirochetes. This is not bacteria, that's part of the villi of the stomach. And that's part of the stomach too. And then here we have bacteria found in the large intestine. This stuff right here is not bacteria. That's actually food or partially digested food. But there's a rod. This looks like a Cuccobacillus. Same thing there, two going through division. And that's a short rod, very short rod. That's either a very short rod or a Cuccobacillus. And that one is almost a conchi. It's probably not, it's probably a Cuccobacillus, but come on, mouse. That one there. All right, any questions about the normal microbiota and the host? The microbiota has been found in some surprising places. I've already mentioned that the adult female bladder has a normal microbiota. This came as a surprise before 2014, where we assumed that the bladder was sterile, that it did not have any normal microbes in it. And part of the reason was when they sample urine, they would get microbes, but they assumed it was either contamination or the microbe had grown up from the urethra to the bladder, which is true. It happens with the normal microbiota. That's usually the way it colonizes the bladder. But my point is that they thought the bladder was sterile, and it is not, at least in females. There is a normal microbiota. They found normal microbiota in human breast tissue. Mother's milk is not sterile. The mother actually puts microbes in her milk. And this is actually inside the breast tissue where the microbes are. That's a little bit surprising because that's inside the body. But I'm going to guess the microbes got on the nipple, which is open slightly to the inside into the breast tissue. And then the microbes colonize the breast tissue. And when the mother makes milk, microbes are put in that milk. It's actually a good thing for babies because they're getting both the mother's milk and the mother's normal microbes, giving the baby the mother's microbes. Any question about any of that? All right, I'm not sure why this one was a surprise, but they found a normal microbiota in the placenta. And this is a unique microbiota, meaning they're unique species that only colonize the placenta. For those of you who do not know, the placenta only occurs during pregnancy. And after pregnancy, the placenta is actually, it's an afterbirth, and it comes out, and I assume the doctors throw it away because nobody usually uses the placenta unless you use it for medical reasons. But the point is the placenta harbors a unique microbiome, and this microbiome is only in the individual when they have the placenta, meaning when they are pregnant. All right, another term you should come to know in this chapter is microbial antagonism. This is where two species of microbes compete with each other. An example of normal microbiota where the normal microbiota competes with the pathogen is when the normal microbiota occupies the niches or the space that the pathogen would try to occupy. For example, if a pathogen were to fall on my skin, I've got lots of Staphylococcus epidermidis and other normal microbes on my skin. And so the Staphylococcus aureus, if it's only one, is not likely to colonize my skin because there is no room for the Staphylococcus aureus on the skin. So it's competing for a location or a niche. They can compete for resources. For example, if Staphylococcus aureus were to fall on my skin, I have other normal microbes on my skin that use those resources. And if they have used all of them in that area, then the pathogen like Staphylococcus aureus cannot get established. So they can compete for resources. The normal microbiota can also produce acids, making the environment too detrimental for the pathogen to get established. With our human skin and the urethra, the normal microbiota make the tissues more acidic than they would be without the normal microbiota. So the normal microbiota on the skin can metabolize sebum and make it more acidic. And that's why our skin is usually around pH 4.5. And that's because the acetic acid is produced, making the skin more acidic. This is also true in the vagina and the urethra, where the environment is made more acidic, as well as the mouth, made more acidic than it would be normally without the normal microbes being there. Furthermore, the normal microbiota can also antagonize a pathogen by producing a bacteriocin. A bacteriocin is a protein that acts like an antibiotic, except it's much narrower in its ability to act than an antibiotic. So, for example, the bacteriocin produced by E. coli, which does produce a protein called the bacteriocin, this protein only will inhibit Salmonella and Shigella. It would not inhibit other microbes growing in the gut, such as Enterococcus faecalis. Does not harm that normal microbe. It would harm Salmonella and Shigella, though. Any question about any of that? All right, so those are a few of the ways that the normal microbiota can compete with the human pathogen. Occupy the niche, compete for resources, produce acids, and then produce a bacteriocin. Probiotics are the consumption of live microbes intended to exert a beneficial effect. For example, when you eat yogurt that has live microbes in it, the yogurt is a probiotic, and eating the yogurt will increase the chance of, let me see if I can remember this. I believe it's Lactobacillus lactis. You will consume it in the yogurt, and then your skin will have more Lactobacillus lactis because you ate the yogurt. And I suppose I should mention that the bacteria in yogurt come in this one end, don't really spread any further than the lips, but then they come out the other end, and then from the other end, the Lactobacillus lactis can then colonize the entire skin. So initially they'll be grown and obviously around the rear, and then expand further, go up to the back, and then expand further, going into the arms and the legs and the head. Anyways, probiotics, the consumption of live microbes intended to exert a beneficial effect. I should mention that probiotics are gaining in popularity in the medical profession in the United States. The normal microbiota establishes a relationship with its host. It forms symbiosis with the host. Symbiosis is just two different organisms or two different populations living together. The normal microbiota does form symbiosis with the host, where the two live together. However, we can break the symbiosis down into mutualism, commensalism, and parasitism. Mutualism is where both the organism benefits. The bacteria, when it colonizes the host gives it a home, place to live, and if the host feeds it, it could get nutrients from the host. Trying to think of an example. Like on a flower, the bacteria can digest the nectar and then get glucose that way. I can't think of an animal example of symbiosis. That's not right. Mutualism, both organisms benefit. That would be like E. coli benefits from growing in your gut because we give it a home, we give E. coli food. E. coli helps us digest our food, so it benefits us. And I don't know if E. coli helps give us B vitamins or not, but part of the normal microbiota of our gut make certain B vitamins for us, like B12. And then we get a significant amount of this vitamin from our normal microbiota living in our gut. And normally we get about a third vitamin B12 from the normal microbiota. Any questions about mutualism? If not, let's move on to commensalism. This is where one organism benefits and the other is unaffected. The best example of commensalism I can think of is each of us has one nematode in our eye. The nematode does not eat human cells, and I assume the nematode in our eyes is consuming microbes that fall into the eye. Anyways, the nematode gets a benefit. He gets a home from us. And then we are unaffected by the presence of a nematode in our eye. So that is a commensalistic symbiotic relationship. Parasitism or parasitic symbiosis is where one organism benefits at the expense of another. So a parasitic microbe like Staphylococcus aureus gets a home, lives on us, and it may get some nutrients from us, like if it kills cells. So Staphylococcus aureus gets a benefit. We, on the other hand, do not get any benefit. In fact, we may get a detriment. But parasitism is where one organism benefits and it is at the expense of another. So when E. coli grows on us, the expense is we could get a skin infection from Staphylococcus, not Staphylococcus aureus, sorry, from the virus. Any question about any of that? Hopefully I explained that well. I'm not sure I'm doing too well. All right, any questions? Let's see how we're doing here. Let's go for a little bit longer. The normal microbiota does not normally cause disease. When it does, we call it an opportunistic microorganism. This is a microbe that can become opportunistic, meaning it can gain access and cause an infection and then get you sick. But an opportunistic microorganism does not normally get you sick. Usually, opportunistic microorganisms happen in individuals with a compromised immune system, such as when their normal microbiota grows out of whack and then causes an infection. Staphylococcus aureus, I did mention, is found in about 20% of Americans on their skin. If we look at the inside the nose and inside the throat, Staphylococcus aureus is actually found on about 50% of Americans. And I've never tested my throat for Staphylococcus aureus, although I did test it for Streptococcus pyogenes, and I don't remember seeing any Staphylococcus aureus. Any question about the normal microbiota and opportunistic pathogens? If not, let's talk again about Koch's postulates. Koch's postulate was used by Dr. Koch to prove that a given microbe causes a given infectious disease. And we use Koch's postulates today exactly to do that, to prove that a given microbe causes an infectious disease. So Robert Koch first proved that Bacillus anthracis causes the disease anthrax, and he wrote this up and then published like 2 years later the steps he used to show that Bacillus anthracis causes anthrax. And these were about what he wrote up. Obviously, he didn't write it in English, but you're getting the point. We have a sick bunny. Dr. Koch says you need to take the microbes from this sick bunny, grow up those microbes in culture, purify them, get a pure culture, and the microbe that you think is causing the disease, you can then inject it into a healthy bunny. If the healthy bunny gets sick, while the bunny is on its side, because it's a sick bunny, oh, the poor little bunny, then that's a good sign that this microbe causes this disease. But Dr. Koch said you need to go further, and that is you need to isolate the organism you first injected into the species. I guess it'd be this one here. You need to isolate the organism you first injected into the species, get it to grow in vitro, and if you do, then Koch's postulate is met that this organism causes this disease. Any question about any of that? I think I'll stop here and I'll continue with Koch's postulate next time. Oh, wait a minute. Maybe I better check the time. We go to 6:50, not 6 o'clock, don't we? Yeah. All right. I'm not stopping. Sorry. We'll stop early. But there are some exceptions to Koch's postulates. Like, what do you do with a microbe that has unique culture requirements and cannot be grown in vitro? If you can't grow it in vitro, you can't originally isolate it and then inject it into a new host. As far as I know, Bartonella henselae is an organism that we cannot grow in vitro. We just don't know what it requires. For the longest time, for over 100 years, they were unable to get the microbe that causes syphilis to grow in vitro. Recently, they have isolated and found out what pallidum requires. That's the name of the microbe that causes syphilis. They have found what the microbe requires, and when they supplied it to a media, they've now gotten syphilis to grow in tissue culture. But for the longest time, like I said, over a century, they were not able to get it to grow in tissue culture. So if they can't get it to grow in tissue culture, you can't run Koch's postulate. So how did you show that Bartonella henselae causes cat scratch fever disease? Or how did they know that pallidum Staphylococcus or Streptococcus? I think it's Staphylococcus. No, that's not right. Well, anyway, pallidum. How did they get it to know that it caused the disease syphilis? Because we've known that for a long time. Same with Bartonella henselae. We've known that it causes cat scratch fever. But how did we know that? How did we do that? It wasn't using Koch's postulate. So what did we do? Anyone have a guess? All right, what they did was they did vast epidemiological studies. And then they watched negative patients who became positive, and they followed them over time, and then later discovered that they were getting these diseases. And some species can cause different disease conditions, like Salmonella can cause salmonellosis and typhoid fever. These are two different diseases. You cannot use Koch's postulate because every case of the disease must give one disease. And that would be something like the human cold. I mentioned before that not only are there different species causing the human cold, but there are 3 different families of viruses that cause the human cold. So you cannot use Koch's postulate to show that a virus causes the human cold. And then there can be moral and ethical reasons for not doing Koch's postulates. For example, scientists have never injected HIV into a patient to then check and see, did this patient develop HIV? I thought I had it someplace, but I'm not seeing it now. So what we did was we did vast epidemiological study, and then we followed these people over time. And if they became positive on their own, meaning they were negative and they became positive, these individuals were carefully assessed to determine if the organism was causing the patient disease. And usually you get Staphylococcus aureus only when it's spilled on the outside of the bottle. It might be a very minor spill. So you follow these patients over time, and if they become positive on their own, then you know this microbe is likely to cause that disease. And that's how we knew that HIV was causing AIDS long before we got a way of, well, actually, with HIV, we don't have a way of getting it to grow because it's a virus. But Bartonella and Celli, we used epidemiological studies to show that it was causing scratch cat fever disease. For some reason, I was getting mixed up on that name. Cat scratch fever disease. All right, some more terms you should know for classifying infectious diseases. A symptom is a change in the body function that is experienced by a person as a result of disease. However, this is subjective. The patient can know about it, but nobody else can measure it. Like when a patient gets a headache, nobody else can measure that person's headache. No one else can know that person actually has a headache. You just have to rely on the patient to tell you that. That's because we have no way of measuring a headache. A sign, on the other hand, is a change in the body that can be measured or observed as a result of disease. For example, when a patient has a temperature of 102, anyone can measure that, and then we would call it a sign, where the body was changed by the result of a test. Syndrome is a specific group of signs or symptoms that characterize a disease, such as the signs and symptoms characteristic of AIDS. Any question about any of this? A few more terms. A communicable disease is any disease that is spread from one host to another. Examples of a communicable disease are measles, tuberculosis, and chickenpox. A contagious disease, on the other hand, is a disease that is easily spread from one host to another. An example of a contagious disease would be COVID-19 and the flu, a disease which easily spreads from one patient to another. Any question about any of that? So all contagious diseases are communicable diseases, but not all communicable diseases are contagious diseases. Any question about that? All right, here's a question for you. Which of these three diseases is communicable but not contagious? Anyone have a guess? You have a 33% chance of getting it right.
Is it tuberculosis?
Yeah, tuberculosis is communicable, but it is not easily spread from one patient to another. You need long-term exposure to pick up tuberculosis. But measles and chickenpox easily spread and are contagious, as well as communicable. So TB is communicable but not contagious. There are only a few diseases that I know of which are communicable but not contagious. Tuberculosis is one. Leprosy is another. A non-communicable disease is one that does not spread from one host to another. For example, tetanus. When a patient gets tetanus, they cannot spread that disease to another patient. Any question about any of that? You could think of an example where tetanus could be affecting a person and then another person becomes infected. Anyone know what that might be? I'm just giving a generality. Tetanus could spread from one patient to another if the person getting the disease were to be a cannibal and then eat a patient who has tetanus. And of course, you'd have to not cook the patient very well because that should kill the tetanus as well. But normally, a non-communicable disease does not spread from one host to another. I won't ask you a question on the final about tetanus spreading from one host to another if the other host is a cannibal. I'm not going to ask that. Okay, let's talk a little bit about the occurrence of the disease. To understand the full scope of the disease, we need details about its occurrence. This means new and old cases. Some diseases are only reported with the new cases, and that is the incidence. Like the flu, we normally report it only as the incidence of flu this year. So incidence, the fraction of a population that contracts a disease during a specific time, and this involves only the new cases only. Prevalence is the fraction of a population having a specific disease at a given time, or one that had the disease in the past. So prevalence is both the old and the new cases. For example, in AIDS in 2007 in the United States, the incidence was 56,300 new cases that year. However, the prevalence in 2007 was over 1,185,000 people. Any question about incidence and prevalence? Sporadic disease is a disease that occurs only occasionally in a population. For example, some people say the typhoid fever in the USA is a sporadic disease spreading occasionally. Other people, though, can call typhoid fever an endemic disease, a disease that is constantly present in a population. An example of an endemic disease is the human cold and malaria, malaria being endemic to the tropics. But when you get very low numbers, such as the case of the incidence of typhoid fever in the United States, the distinction between an endemic disease and a sporadic disease is that there's very little distinction. And that's why some people call typhoid fever a sporadic disease, and other people say, no, it's always present, especially among the homeless population. Typhoid fever is an endemic disease. The others, like an epidemic, we have a good definition for how this differs from an endemic and a sporadic disease. An epidemic is a disease acquired by many hosts in a given location in a short period of time. And to get an epidemic, you need to have significantly higher numbers of patients than you expect. And that's an epidemic. An example is influenza. About every 7 to 10 years has an epidemic. And that's mainly because it's a new variant and our host immune systems aren't ready to face the new variant. But the eyes and the limbs will be working like within the first 2 weeks to make antibodies. And then they'll make antibodies which will go out and attack the pathogen. A pandemic is an epidemic that becomes worldwide. For example, COVID-19 was initially in epidemic status in China. However, once it spread beyond another continent, it is then a pandemic. So if it's ever a disease spreading to 2 or more continents, it is a pandemic. And it usually becomes worldwide. I can only think of one case where we had an epidemic that became pandemic, spreading to only 2 continents. No, it was actually 3 continents, and did not become worldwide. And that was the occurrence of SARS back in 2003. Any question about any of that? Another useful way of talking about disease is discussing the severity or the duration of a disease. An acute disease is a disease where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. The human cold would be a sporadic disease where you initially get infected, short time later you get so that you're producing the virus and can spread, but you eventually either melt down the tip or you broke the tip, and then you cannot be stabbed by the ruler. Actually, you'd be very unlikely to be stabbed by a ruler anyways, but if it's face up, you would be much less likely to be stabbed. A chronic disease, on the other hand, is a disease that develops slowly, persists for a long period of time, and generally gets worse. It doesn't always get worse, but it generally gets worse. An example of a chronic disease is mononucleosis, where initially the patient is just slightly sick, and maybe several months later, their number of the viruses will increase. Let me get paint out so I can show you that. Not much to show, but so the last one, chronic disease, is one where the disease starts slowly, the symptoms are very mild, and then over time, the number of microbes increases slowly until at the very end where there may be a bloom. I'm not going to break it because the patient died. So normally this is something seen in a hepatitis B vaccine inoculation. Now I lost what I was going to draw. Anyways, that's a chronic disease, a disease that starts slowly and then gets worse over time. However, for that disease, we also have, how do I get out of here? something like this. Let's extend it. And if the number of microbes never increases beyond a certain amount, like right here, come on, mouse, never increases beyond right here, this just plateaus out. We'll take that way to the back of the thing. So make that a straight line. I can't draw perfectly straight, but this would be a persistent disease or a chronic disease the patient tolerates, meaning the patient does not die, doesn't bloom and then kill the patient. An example of a chronic disease is herpes, Galeas, I guess, where the microbe can grow on your skin and can increase in your skin, but it does not get so numerous that it starts doing damage to the skin. Any question about any of that? The point is it can be tolerated, in which case the graph will look like that. There is a disease category between acute and chronic disease. I've got it highlighted because you do not need to know it. It's called subacute disease, where the symptoms are between an acute and a chronic disease. An example of a subacute disease is subacute sclerosing panencephalitis. Symptoms between an acute disease and a chronic disease. Okay, let's go on just a little bit further. Let's talk about the extent of host involvement. Infections can be classified according to the extent which the body's host is affected. A local infection is where the pathogen is limited to a small area of the body, such as when the patient has a boil. It can be caused by Staphylococcus aureus. The Staphylococcus aureus is not causing boils all over the skin. And so the pathogen is limited to a small area around the boil. Another case of a local infection is an abscess. So that's a localized infection. We can have a systemic or generalized infection, and this infection occurs throughout the entire body. Like when a patient gets infected with measles, measles will be made in many different tissues in the patient, and then the virus will spread to many different tissues, potentially all different tissues. And then there's a focal infection. This is a systemic infection that began as a local infection. For example, MRSA, multi-drug resistant Staphylococcus aureus, can be growing on the skin and causing a skin lesion. At the moment, that is a local infection only. However, it can become a focal infection when it spreads beyond the skin. Like if the Staphylococcus aureus invades the tissue, it can invade all the way down to the blood and then put Staphylococcus aureus in the blood, which can then circulate in the body. Any question about any of those terms? Some more terms. Bacteremia is the presence of bacteria in the blood. You should compare that to septicemia. This is the growth of bacteria in the blood. The only difference is one is growing, the other, as far as we know, it's only present. We suspect it's probably growing, but most clinicians, when they get a microbiological test ordered and returned to them, they would only say septicemia because most of the tests do, excuse me, not septicemia, they'd only say bacteremia because most of the tests do not look to see if the bacteria is actually growing in the blood. They'll just see it in the blood and call it bacteremia. Septicemia, you need specific tests. Like if you were to look at this later, and if it were to expand in numbers, then you could say it's septicemia. But from one test, at least looking at the microscope, you can't tell. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. The state of the host involvement in an infection also depends on the host resistance, meaning the host immune system can defend the host. A primary infection is an acute infection that causes the initial illness. For example, the child is healthy, and then the next day, the child has a sore throat. The sore throat is the primary infection. A secondary infection can then happen after the primary infection predisposes the individual for a secondary infection. For example, a child has a sore throat and a cold, and later they get an ear infection because the ear infection was an opportunistic organism that infected the ears and then grew there, causing the secondary infection. A secondary infection usually needs a primary infection to damage the host or weaken the host to make the secondary infection possible. There's also a subclinical disease, and we've discussed it before. It is an asymptomatic disease, an inapparent infection, where a patient has no noticeable signs or symptoms, and yet they do have the disease-carrying organism that is growing in them, and they're shedding these disease-carrying organisms. A good example of a subclinical disease is COVID-19, where about 50% of young people, young adults, teenagers, and children have no signs or symptoms of getting or having COVID-19. And this is a subclinical disease. Any question about any of that? All right. I've already talked a little bit about a latent disease. This is a disease where the patient has a period with no signs or symptoms of the disease. For example, a child comes down with chickenpox, and then maybe decades later, that virus comes out and activates. We call that a latent disease, where the patient has signs and symptoms of disease once again. It's just that the signs and symptoms aren't the same as the chickenpox, but it's the signs and symptoms of shingles. And you don't need to know why, but what happens is the virus actually is hiding out in the ganglia cell, which controls a certain region of the body, meaning it innervates a certain region of the body. And then when the virus activates, enters the budding life cycle out of the latent life cycle, the virus can go down the nerve, and then every region of the body which is innervated by that one nerve can have shingles in that region. So for example, for the upper arm, the shoulder, and the very upper part of the back, that would be one area of the body which is innervated by one ganglia. And then shingles can grow down that nerve, and then you have shingles all in that region. Different regions of the body have different ganglia that control them, and usually with shingles, you only have one ganglia that activates at a time. Any questions about latent disease? Remember, in latent disease, the pathogen is inactive while the pathogen is in latency. All right. The last topic I want to talk about is herd immunity. This is when many immune people are in a population. They can protect the herd, meaning protect the non-immunized people from getting disease, because the disease cannot spread to an immune person. Now, initially, if nobody is immune, they're all blue, susceptible, and then red would be a contagious individual. Initially, most of the population would then become sick from this one agent. So only some very lucky individuals do not get sick. When you have herd immunity, this is where a large number of people are immune to the disease. Now, when we have our two individuals who are contagious, only the people who are not immunized, being blue, who are very close in contact with the contagious individual, will become sick of the disease. So instead of almost all the population becoming red, only two other members of the population will become red. And why is because when the organism leaves this patient, the red patient, and goes to another patient, these individuals are all immunized except for that patient. And so none of these immunized get infected, and then the disease can't spread from like this patient to that patient. That will protect this patient by herd immunity. Now, this patient is not protected because they're too close in contact with this patient. And so they will get infected and come down with disease. However, the disease is unlikely to spread from this patient because it needs to go through an immune patient, and the disease can't spread in the immune patients. And that's how herd immunity can protect the herd, where the many immune people prevent the spread of an infectious population throughout the individuals. Most will not get infected if herd immunity is in place. For some diseases, you need to have the immunity very high, or the disease can spread. Measles is a case in point, and I think I read that measles was actually spreading somewhere in the United States. It might have been Oregon, but I don't remember. And what happens is you need 90% of the children to be immune to prevent the spread. And if it's less than 90%, then the virus can get out of this individual, infect a non-immune individual, and then infect another non-immune individual, meaning the herd immunity isn't high enough. With measles, you need over 90% of the population to be immune. All right, any question about anything? Oops, didn't mean to go there. If there's no question, I'm going to stop here, and I will log in at 7 o'clock to answer questions in the lab, but Nobody needs to show up if you do not have questions. And I'll log out at around 7:15. Any questions about anything before I go? All right, if not, I'll say goodbye and I'll see you next time.